Thank you so much, everyone, for taking the time out and joining today's Creative Biolabs live webinar series. My name is Renee. I'll be your moderator for today. First, please allow me to briefly introduce our company, Creative Biolabs. Creative Biolabs is a leading contract research organization enthusiastically exploring immunotherapy discovery with unrivaled expertise, substantial resources, and also hands-on experience. Creative Biolabs has established a novel cell therapeutics platform specifically serving clients working on cellular therapy research. We can provide various products ranging from CAR constructs, TCR products, and diversified kits to other innovative solutions, including target identification services, CAR construct, and CAR T in vivo and in vitro evaluation. The theme of today's webinar is engineering strategies for improving adoptive T cell therapy against cancer. We have brought the seminar to you for the below reason. The adoptive cell transfer of tumor-specific T cells has emerged as a potent treatment against some advanced cancers. Adoptive cancer transfer with peripheral blood T cell gene modified to express chimeric antigen receptor T cells targeting the B cell lineage antigen CD19 has led to complete remission for up to 90% of some advanced treatment refractory hematological cancer patients. However, unprecedented response to CAR therapy have not been achieved against epithelial-derived solid tumors, by far the most common category of cancers. Indeed, solid tumors present a variety of barriers to CAR therapy, including that there's a paucity of tumor antigens not also found on healthy tissues, thus running the risk of on-target but off-tumor toxicity. We have invited Dr. Melita Irving to join us in this webinar section and discuss the latest findings of her research team on innovative CAR design, hoping to provide insights into the engineering strategies for improving the efficacy and safety of CAR and TCR cell therapies against cancer. And now on to our distinguished speaker today, Dr. Melita Irving, who first worked in the field of T cell engineering for cancer immunotherapy in Switzerland back in 2007. This is now the group leader at Ludwig Cancer Research in the Department of Oncology at the University of Lausanne since 2013. Dr. Irving's team focuses on the development of tumor-directed TCR and CAR T cells that are programmed to logically respond to microenvironmental cues and small molecule administration for maximized function, fitness, and safety. Her team has optimized retroviral, lentiviral, and CRISPR-Cas9-based tools and methodologies, as well as T cell expansion conditions, allowing efficient evaluation of next-generation T cells in the context of both syngeneic and xenograft tumor models. So before Dr. Irving starts her actual presentation, I just want to quickly remind our audience today that this is an interactive webinar. So if you have any questions during this webinar, please type your questions into the Q&A panel, and Dr. Irving will run a live Q&A session and take as many questions as time permits at the end of the seminar. Also, for those questions left unanswered, we can arrange to resolve them offline. So without further ado, let's welcome Dr. Melita Irving. And Dr. Irving, you should be able to share your screen and begin whenever you're ready.
Thanks, Renee. I'll start sharing now. Okay, good morning, everybody. Oh, can you hear me now? Can you hear me?
Yes, we can hear you.
Okay, great. Good morning, everybody, and good afternoon from me. I'm located in Switzerland. First of all, thank you very much to Renee for the very nice introduction, and to Creative Biolabs for the invitation to present today on the work that we're doing on the development of engineering strategies for improving adoptive T cell therapy against cancer. So adoptive cell therapy of cancer, there's two main forms. The first is to take a patient biopsy and expand out the tumor infiltrating lymphocytes that are specific for the tumor and then reinfuse them into the patient. Alternatively, you can take the peripheral blood T cells from the patient and then engineer them to express either a T cell receptor recognizing the tumor or a so-called chimeric antigen receptor. And as Renee mentioned, there's been tremendous success for some of the hematological malignancies with CAR therapy. However, there we go. However, the same success has not been met today for the treatment of solid tumors. And why is that? Well, there's many barriers. For example, there can be limited homing and engraftment of the T cells, so to reach the solid tumor and graft within that tumor. Also, once the tumors do reach the solid tumor, they face many barriers. It's a very hostile microenvironment for them. In fact, there's usually limited oxygen and nutrients that they need for their functionalities. The pH is typically low, so it's acidic. And there's a range of inhibitory molecules and receptors, some of them brought in by inhibitory immune infiltrates, such as T regulatory cells or myeloid-derived suppressor cells. And this together can lead to energy or exhaustion of the T cells, their limited persistence, and eventually failure of the treatment. Another important issue is the fact that there's very few bona fide solid tumor antigens that are only found on tumor cells and not also found on healthy tissues that can in fact lead to important toxicity in patients. So how can we overcome some of these barriers? Well, there's many approaches that can be taken. For example, and many of these topics we're addressing in the lab, it's possible to optimize the binding properties of either the T cell receptor or the CAR design, also the affinity of the single chain Fe, or the design itself, which I'll come to later in the talk. And this can be done to increase both the specificity and the safety of the receptors. Another important concept is developing optimized activation, transduction, and expansion conditions. So you want to, be able to expand enough T cells, but you also don't want to terminally differentiate them. You want them to be less differentiated, more of a central memory type phenotype, such that they're better able to expand and engraft in the patient. Now, it's also possible to implement co-engineering strategies. So you have the CAR or the TCR plus accessory molecules or receptors, and these can be designed either to directly promote the function of the T cells themselves, and also harness the endogenous immune system and reprogram the tumor microenvironment. We also believe that it's very important to perform robust preclinical testing in syngeneic tumor models or humanized ones, because it's really important to understand the efficacy of the therapy in the context of a fully competent immune system, which can either serve to support the therapy or hinder it. And this also offers the opportunity of looking at potential toxicities. And then lastly, this doesn't have to do with engineering, but it is possible to implement combinatorial approaches such as classic approaches such as radiotherapy, chemotherapy, immune checkpoint blockade. Again, this can be to support the TCR, the CAR T cells themselves, and promote endogenous immunity. So I'll talk about a few of these topics. The first topic I'd like to talk about is engineering safety and efficacy into CAR design. So I'm just going to discuss briefly a stop CAR paper that we published a couple of years ago. This work was largely done by Greta, Elise, and Elisabetta from my group, and also our collaborators who I'll mention later. So before I move on to the stop CAR concept, just to briefly remind you about CARs. So CARs typically comprise the single chain Fe. So this is what recognizes the tumor antigen. This can also be a ligand, for example. And the first generation CARs only comprise the endodomain of CD3 zeta. So this provides signal 1 of T cell activation. So it enables the T cells to become activated when they engage target antigen. Now, the first generation CARs largely failed in the clinic, and it was noted that there was very poor persistence of these T cells, and possibly this was due to a lack of co-stimulation. So then came second and third generation CARs, which simply mean that they also comprise one or more co-stimulatory endodomains. Now, most CARs that are used in the clinic have the co-stimulatory endodomain of either CD28 or 41BB, and typically it's second generation CARs that are used. And now we're moving more and more into so-called next generation or 4G CARs. And this simply means that the T cells are engineered not only with a CAR, but also with an accessory receptor or molecule, again, to either directly support the function of the CAR T cells or to harness endogenous immunity. Now, various designs have been developed to improve the safety and function of CAR T cells. For example, in the classic split CAR T cell approach, T cells are engineered with two receptors targeting two different tumor antigens. And you can see that on one of them, you have CD3 zeta endodomain for T cell activation. And on the second one, you have the co-stimulatory endodomain. So what this means is that both of these tumor antigen targets need to be co-engaged for full activation of the T cells. Another approach is the so-called on-CAR. Again, the idea is to split the signaling, the CD3 zeta endodomain and the co-stimulatory endodomain on two receptors. Here in their native state, these two receptors are not bound together. So if the CAR T cells encounter antigen, nothing will happen until you provide a small molecule that essentially heterodimerizes these chains, bringing them together. And then in the presence of target antigen, the CAR T cells can become activated. Another approach is to develop a so-called ICAR, so you could have a single chain Fv that's directed against a healthy tumor antigen. You don't want to activate the T cells, and you can connect it to an inhibitory endodomain, such as from PD-1. So we, with our collaborator, Professor Bruno Correa from the EPFL, also in Lausanne, came up with the concept of a so-called stop CAR. And so similar to the split CAR and the on-CAR, we envisioned having two chains, a receptor chain or the tumor antigen binding chain and a signaling chain. But here in the native state, opposite to the on-CAR, we envisioned having the two chains bound together by a so-called chemically disruptible heterodimer in the native state. And then with the application of a small molecule that could disrupt this protein interface, we would separate the two chains and thereby abrogating CAR T cell activity. So this beautiful modeling work was done by Professor Correa and his group. And so to develop the chemically disruptible heterodimeri, we started with several criteria. First of all, we wanted to work with proteins of human origin with minimal deviation from wild type sequence. This is so that there would not be immunogenicity, hopefully, of these constructs in patients and their T cells. We wanted to work with proteins that were well packed, also ones that would not interfere with synapse proximal T cell signaling, and ones for which there's a small molecule that could be commercially available, approved for use in patients, having a long half-life and also well tolerated. And so when we started the study, we identified the BCL-XL interaction with the BH3 domain BIM as a promising starting point because there are several clinically relevant anti-apoptotic drugs already developed. However, the binding interface between BH3 BIM and BCL-XL, the BIM itself is unstructured. So what we wanted to do was to be able to identify essentially a globular human protein scaffold onto which the critical binding residues could be engrafted. And so I won't go into detail about the modeling because this is not my specialty, but essentially they use motif graph to screen up protein structure databases. And they identified three lead target proteins onto which they could engraft the critical binding residues. And lead design 3 was what we moved forward with. It was the protein apolipoprotein E4, and we engrafted hotspot residues onto it and several others, leading to a picomolar binding affinity, in fact. And they showed beautifully in the lab in vitro that the two proteins, BCL-XL and this chimeric fusion protein, could be disrupted by a small molecule. Then in my group, we wanted to build this into a CAR. So again, we wanted to develop two separate chains. We went through several iterations of the signaling chain before we had one that was sufficiently self-surface expressed. We did our initial screening in Jurkat cells, or Jurkat reporter cells, in fact. And by amnest imaging, we did have some indication that probably the two chains were associating in the cell membrane by overlap. And we showed that in a reporter cell line that they could be activated equally well as an equivalent second generation CAR. So we developed this CAR to bind to PSMA, and we showed that there was good expression and good stability in Jurkats. And then we moved to human T cells. Again, we were able to show self-surface expression of both of the chains, lower than the second generation CAR, but still high enough for functional assays. We showed that the fold expansion was fine, that in fact, even up to day 30, we could still detect CAR on the surface, and the CAR T cells were actually still functional at that time point. And in functional assays, we showed that they efficiently killed target PSMA positive tumor cells equally well as second generation equivalent CAR T cells. Of course, what we really wanted to know was, could we disrupt the functionality of those stop CAR T cells in the presence of a small molecule? And we showed that this was in fact the case. So here I'm showing an incucyte assay. As the curve increases, this means that the target tumor cells are being killed. So you can see that the stop CAR T cells efficiently kill target tumor cells. However, in the presence of small molecule, this is abrogated. Whereas for the second generation CAR T cells, it doesn't matter if the drug's there or not, it doesn't impact the CAR T cell. And then, of course, most importantly, we wanted to have the stop CAR function as a more of a rheostat. So we did not want this to serve as a suicide switch that you apply the small molecule and you lose your very expensive CAR therapy. We wanted to have an approach that was reversible. And we showed that this was in fact the case. So again, this is a killing assay. The stop CAR T cells alone kill. In the presence of small molecule, they do not. And what we did was we took stop CAR T cells, we pre-exposed them, we stopped their functionality, and then we arrested them and we re-exposed them to target cells, and they could fully regain function, killing and cytokine production. So we were very happy about this. And then finally, even more important, I would say, is the in vivo functionality. We did show that for the stop CAR T cells, they could equally efficiently control PC3-PIP subcutaneous tumors as second-generation CAR T cells. And while second-generation CAR T cells, their tumor control was not blocked in this presence of small molecule, the stop CAR T cells lost their ability to control tumors in the presence of small molecule. And then finally, we did show the dynamic in vivo control of stop CAR T cells based on a tough reviewer. And essentially what we showed was that if you apply the stop CAR T cells in the presence of drug, they're not controlling tumors. You stop administering drug, they begin controlling, and vice versa. If you apply them in the presence of drug, there's no tumor control, you stop giving the drug and they start controlling the tumors. So we were very delighted with these findings. So that ends the part about the stop CAR. So I'm going to talk about a few different stories. So they're different topics. I previously mentioned how we feel that it is very important to evaluate T cell therapies in the context of syngeneic tumor models so that we can really understand the efficacy of the therapy, especially for next generation CAR T cells or combinatorial treatments, how the local tumor microenvironment impacts their outcome. And this work was largely done by Everest, Georgia, and Paris. So when we started this work, we wanted to work with mouse T cells, C57 black 6 mice. And to begin, we had very, very poor transduction efficiency. And we put a lot of effort into optimizing our methods. We published this work last year in JexMed. I won't go into all the details, but we did publish very, very detailed methodologies for our transduction and expansion of marine CAR T cells. A few important steps is that we do concentrate our retrovirus, and then for the expansion, we do expand them in the presence of starting in IL-2 for a day or two, and then expanding them further in IL-7, IL-15. So the importance of expanding the cells in IL-7 and IL-15 is because we want to generate less differentiated T cells. And there's already evidence in human T cells that if you do expand them in 7 and 15, they don't drive terminal differentiation like IL-2 does. You have more of a central memory type phenotype. This is what you want because the T cells have much better engraftment in the patients or in the mice, and you get much better tumor control. This has been well documented now. So in summary, for our optimized protocol, we had better cell surface expression, we had less differentiated T cells, we had, so here using the surface markers CD62L and CD44L, you can see we have almost 64% central memory versus 43% if they're fully expanded in IL-2 only. We had better fold expansion in the presence of 7 and 15, higher viability, and actually also higher in vitro function of the CAR T cells expanded in 7-15. So then based on our really excellent transduction efficiency with our protocol, we routinely achieved 75-80% transduction efficiency for the CARs. We next moved on to co-engineer the marine CAR T cells. So I previously mentioned when you can co-engineer T cells, it could be, for example, to secrete decoys of PD-1 or soluble molecules that can activate the T cells or other T cells in the immune system. It's also possible to knock down genes, inhibitory genes, for example, and to further increase safety, what we often do is we do encode these genes under an inducible promoter such as NFAT, such that they're only expressed in the context of activated T cells in the tumor microenvironment. And so for this work, we did see very nice expansion of T cells in the presence of 7-15, and then further inspired by work in human T cells, we decided that we'd co-engineer the CAR T cells with interleukin 15. Again, with our protocol, we achieved very high transduction efficiency, and we could show that IL-15 was nicely co-expressed by the CAR T cells. These T cells that were co-engineered to express IL-15, they had a better phenotype, so there was a higher proportion of Ki6 positive, BCL2 positive cells. We also had a very nice central memory phenotype of the cells, again, better fold expansion, and a superior cytokine production as well. So there were definite in vitro benefits. And so then what happens in vivo? Indeed, we saw that in vivo, this is a B16 tumor model. We did see significantly improved tumor control with IL-15 co-engineered T cells. And we saw that there was a higher level of infiltrating 4G CAR T cells than 2G CAR T cells in the tumor. Moreover, they had a better phenotype in vivo. So the IL-15 co-engineered, they had lower PD-1 expression. We saw this both in vitro and in vivo. And moreover, we did see a reprogramming of the tumor microenvironment upon transfer of the 4G CAR T cells. So we saw higher levels of activated NK cells and fewer M2 macrophages, and both of these would favor tumor control. So in summary, we saw very excellent in vitro benefits of IL-15 co-expression, which also translated in vivo better persistence of the cells, better proliferative capacity, lower PD-1, superior tumor control, and favorable reprogramming of the tumor microenvironment. So for that project, we were actually working with a VEGF-R2 directed CAR T cell. So this moves now to the concept of tumor antigen targeting. And so there's a lot of interest in actually targeting the tumor vasculature by therapies, because tumors are highly reliant on blood vessels for receiving nutrients, expelling waste, and for their growth. And so there has been some work on CAR T cells developed against tumor vasculature antigens, which also have the advantage of being more accessible, so the T cells don't need to enter into the tumor microenvironment. And in principle, there should be higher stability of the antigen targets as well. However, we also know from some of the seminal work of George Kukos and others that there are also barriers within the vasculature itself. There can be suppressive molecules and receptors. So we've been working with this VEGFR2 CAR T cells, and we typically saw that as a monotherapy, we saw no tumor control. And we knew that from an in vitro, that really it's a very potent CAR. They, you have high effector function of 2G CAR engineered marine cell T cells. And so we wanted to really understand what was going on there. Could there be barriers in the vasculature that could be overcome to improve their efficacy? But of course, first we wanted to look to see if there was downregulation of the target antigen VEGFR2. And we found that this was not the case. There were similar levels of VEGFR2 expression pre and post ACT. However, we did notice that there were higher levels of VEGF-A present post-treatment. And so this led us to question, could the CAR T cells actually be physically impaired by VEGF-A binding to a shared binding site on the VEGFR2 receptor? And so I walked down the hall to talk to a collaborator of mine, Professor Vincent Zoit, and he did some homology modeling for us. And there was every indication that, in fact, yes, VEGF-A, where it's binding to VEGFR2, could in fact be impairing the binding of our single chain Fv from engaging the target. So we set up a series of in vitro competition assays. And indeed, we showed that if we co-cultured CAR T cells with VEGF-A, we could impede the CAR T cells from binding VEGFR2. And then we set up functional assays as well. We showed that we could block the adhesion of anti-VEGFR2 CAR T cells in the presence of VEGF-A. You can see a drop in adhesion here, which could be rescued in the presence of anti-VEGF-A antibody. And we also showed that there was lower activity of the CAR T cells in the presence of VEGF-A. So we then went to move on, and we questioned, could we apply anti-VEGF-A antibody to improve tumor control by the CAR T cells? And indeed, we did see that this was the case, that we saw superior tumor control upon transfer of the anti-VEGFR2 CAR T cells and anti-VEGF-A antibody. Now, of course, anti-VEGF-A antibody could have other positive effects on the tumor microenvironment because VEGF-A, of course, is associated with MDSC accumulation and the induction of Tregs, and DC maturation. So if you block it, you would impair these negative factors from happening. And we did see that there was a favorable impact on the infiltrating CAR T cells in the presence of anti-VEGF-A antibody as well. And then finally, this is not about T cell engineering, but I just really briefly wanted to mention the whole concept of using rational combinations to support T cell control of tumors. So this is the work largely of Fernanda Herrera and Catherine Renee from my group, previously from my group. And we had, so low-dose radiotherapy is commonly used in the treatment of cancer, but typically at higher doses, and it directly kills target tumor cells. However, there was some very inspiring work showing that even at lower doses, it could have an important effect on modulating the tumor microenvironment, activating dendritic cells, releasing danger signals, upregulation of cytokines and chemokines, and normalizing of the vasculature. And so we were really interested in the treatment of advanced ovarian cancer, which traditionally high doses of irradiation had been used for treatment, but it, of course, is highly toxic. We wanted to understand if low-dose radiation could be used to reprogram the tumor microenvironment and make it responsive to immunotherapy. And so we explored low doses, 0.5 gray, 1 gray, and 2 gray, and we were using the ID8 ovarian cancer model. And so essentially what we started with was just looking to see what would happen if we took advanced, so engrafted mice, these are IP engrafted tumor cells, and just applying low doses of irradiation. And then we use nanostring to look at were there upregulated druggable targets. And in fact, there was a massive reprogramming of the tumor microenvironment, including PD-1, CTLA-4, CD40. And so we came up with a rational combination of treatment that we called RACIM. So radiotherapy, cyclophosphamide to get rid of Tregs, and immunotherapy. For the immunotherapy, we used a combination of anti-PD-1 antibody, anti-CTLA-4 antibody, and CD40 agonist antibody. And we treated the mice as if we'd be treating patients. We treated them in 3 cycles. At each cycle, they received the radiation, the cyclophosphamide, and immune-stimulating antibodies or the immune-blocking antibodies. And we showed that with the full combination of RACIM, we had a very potent effect in terms of tumor control and survival. But very interestingly, if we pulled out any of the components of the treatment, we did lose efficacy and survival. And very interestingly was the fact that if we pulled out the radiotherapy at any of the cycles, we also lost efficacy at the therapy. So radiation at each cycle was very, very important. And this was associated with an influx of immune-infiltrative T cells. And we did go further. We went and did single cell sequencing. I won't go into these data, but they are published. But we did show a massive reprogramming of the T cell compartment and also of the myeloid compartment. And so, of course, we're very interested in applying low doses of irradiation to support CAR T cell therapy. And there is some preclinical work suggesting that this could be a very positive combination. So thank you very much for your attention. I know I covered a lot of topics, but I find all of this very interesting. And I really think to be able to really tackle solid tumors, we do need combination approaches. We need to understand the mechanism of escape of tumors and how can we optimize T cells through engineering, culture expansion, et cetera, combination therapies. So we have developed a stop CAR that we showed that we could remotely and reversibly control in vivo. We've optimized retroviral transduction of mouse T cells and shown best expansion in the presence of 7 and 15, allowing really comprehensive in vivo studies in the context of a fully competent immune system. And that does, of course, allow you to interrogate the immune system and understand what are the mechanisms of resistance and then go and further engineer T cells. We showed that IL-15 co-engineering improves CAR T cell fitness, both in vitro and in vivo, allowing enhanced tumor control. And that, in fact, IL-15 co-expression, CAR T cells also does reprogram the tumor microenvironment. We were able to show that because we were working in a syngeneic tumor model. We also looked at VEGFR2 CAR T cells and showed that they were being, this particular CAR T cell with the DC101 single chain FE, that it was being physically blocked by VEGF-A, and we showed that we could rescue it with anti-VEGF-A antibody. And finally, I briefly discussed our work on low doses of irradiation to reprogram the tumor microenvironment and allow responsiveness to combinatorial immunotherapy. Thank you very much for all of your attention. I'd like to acknowledge several very important people, of course, Professor George Kukos, the director of the High Tide, as well as Ludwig Cancer Research at the University of Lausanne and the Department of Oncology at the CHUV Hospital. Thanks to him for all of his leadership in making these projects possible. Thanks also to the different former and current members of my group who've been involved in these different projects, who I've listed throughout the talks and are very important collaborators. And finally, thank you very much to all of the funding resources, in particular Ludwig Cancer Research, the Swiss National Science Foundation, the Prostate Cancer Foundation, OncoSuisse, Cancera Isric Foundation, the ERC, and the Bill Temme Foundation. Thank you again for your attention.
Thank you so much, Dr. Irving, for this great presentation.
Thank you very much. It was my pleasure.
I hope our audiences enjoyed it as much as I did. I hope that we do have some questions I see.
Okay, great.
Let me see if I can get my camera. Come to the side. Okay, perfect. There we go. All
Oh
right.
yeah, there's a question about yes, I know that a recent paper has come out looking at targeting angiogenesis in the eyes. I'm not sure if they were using CAR therapy. I think they were knocking out VEGF-A, if I'm correct. CAR T cells might be a little bit dangerous to apply to the eyes, but thank you for that question.
Okay, we have another question from Alexis Cockcroft. They said, am I right in thinking that small molecule disruption is intended to be deliberate strategy for modulating CAR T function and would be complementary to the use of tocilizumab or similar for specific adverse reactions such as cytokine release syndrome?
Yeah, exactly. So we do envision the stop CAR T cell to be a way of modulating or fine-tuning CAR T cell response levels. So if you have an early indication that the patients are having a very high response to the therapy, you could apply the small molecule and tone down the activity of the CAR T cells. And of course, you could give anti-IL-6 antibody if this is not enough, just slowing down the stop CAR T cells. So we kind of envision the stop CAR T cells, it could be against known tumor target antigens, where it may be difficult to evaluate the level of antigen expression in the patient, like how, for prostate cancer, for example, how disseminated is it? You could tune down the functionality of your anti-PSMA CAR T cells this way. And then the on-CAR might be more desirable for going after new targets where you know less about potential on-target but off-tumor toxicities that could occur.
Awesome. Okay, so another question. In terms of TCR discovery and validation, what special technologies are you using to validate TCR affinity and avidity? Are there assays you find extremely useful?
Oh, for TCRs. So yeah, so actually back when I was in the lab, I was doing ViaCore. So I was refolding soluble T cell receptors and evaluating kinetics, on rates, off rates, and affinity using the classic ViaCore approach. We're now also looking in terms of avidity. We're using something called NTA tetramers. So essentially you take your TCR T cells and you bind your NTAmers are called. So it's essentially multimeric peptide MHC, and the peptide MHC are essentially on a scaffold. So it multimerizes them. You apply imidazole, the scaffold releases, and then you have essentially monomeric peptide MHC remaining, which is labeled with a fluor. And then in real time, you can look at the dissociation of those monomeric peptide MHC complexes to get an idea of avidity this way, or off rates on T cells directly for the T cell receptors.
Nice.
Yeah, there's other approaches too. There's dextramers. There's really several interesting products in the market.
Excellent. Okay, another question we have. Can you do a comparison between syngeneic and xenograft tumor models and CAR T studies? Is the syngeneic model more preferred to use in CAR T studies involving mice? And are there any differences in the lab setting?
Okay, that's a very complex question. So I think good question. Thank you. So I mean, we don't always have the equivalent CARs that we have for the human setting in the context of the mouse. The advantage of the syngeneic mouse is really being able to explore the tumor microenvironment. And also you can look at potential toxicity of the single chain Fe. So if you have a signal chain Fe targeting molecule X, and molecule X is also found on various healthy tissues, you can see if there's toxicity against those tissues in the mouse in this context. Whereas in the xenograft model, you essentially engraft a tumor. Mostly we're working with subcutaneous, and it's typically a human CAR. Single chain Fe are not necessarily cross-reactive with the mouse antigen, so you won't see the effect of toxicity, and you won't really understand suppressive mechanisms that could take place in the context of a fully competent immune system. So, but it's important also to evaluate in the context of human T cells. So I think you really do need both for your studies. I hope I've answered everything that was asked in that question.
I guess a part of it is the syngeneic model more preferred to use in CAR T studies involving mice?
I guess I think you need both. I think it's really important in terms of the, in the context of an endogenous immune system and toxicity. In that way, it's favorable. But you also, if you're going to go into humans, you do want to be testing human T cells. And there's a lot of people doing really great work with humanized mouse models as well.
Good. Okay. Another question.
Wow.
Successful transduction and expansion of murine CAR T cells are proved to be very challenging.
Yes.
And we know you're doing experiments in your lab. Can you share some firsthand experiences with this?
Yeah, so the paper that I mentioned, our Journal for Experimental Medicine paper, we really published all of the details of our protocol for virus production, transduction, the purification of the mouse T cells, the activation, the transduction, and their expansion. And all of the details are in that paper. But of course, you can always reach out to our lab if you have further questions. Or since we published that paper, we've had a lot of people reach out to us for help. So we're really happy that it's helping other people. Yeah.
Good. Okay. Next one. We noticed emerging studies around CAR macrophage, which seems like a promising immunotherapy candidate to target solid tumors. What is your opinion on this? And would you anticipate its prevalence in immunotherapy?
Yeah, I mean, it's actually really exciting, the work that's been done with macrophages, because a lot of tumors are very highly infiltrated by macrophages, and some tumors are quite T cell excluded. So it can be very difficult for the TCR T cells or the CAR T cells to penetrate them. So you could imagine also co-engineering macrophages and transferring them into patients to sort of reprogram the tumor microenvironment for T cells, or engineering them with a CAR, as has been done. I'm pretty sure Carl June has evaluated macrophages engineered with CARs, and it's very promising, I think.
Excellent. Good. Okay. Actually, I have a follow-up from the first question about the CAR T function and tocilizumab. They asked, if I understand correctly, if I understand the CAR correctly, could the CAR T against VEGFR2 be used for treatment of eye conditions? Oh, that's when you looked up. Yes. Okay.
Yes.
Okay. Perfect. Okay. I think that is all of the questions I have so far. I think that's it for now.
That's great. No, really nice questions. That's too bad I can't see who's out there in the audience, but thanks for joining us this afternoon, and hope you enjoyed it. And don't hesitate to reach out if you have technical questions.
Definitely. So please note that Creative Biolabs regularly invites brilliant experts such as this in the fields of science and biotechnology to present their most recent findings in our webinar series. So please visit our website and subscribe to our social media so you won't miss out on any upcoming webinars. That's all for today. And thank you everyone for time again. And we'll see you next time. Thanks.
Thank you, Renee. Thanks also, Anna. Bye-bye.
Thank you, Dr. Melita. Thank you.
Bye.
Bye.